Imagine you are trying to build a family tree for every single person in a massive, sprawling city like, I don't know, New York or Tokyo.
Right, a total logistical nightmare.
Exactly. But you don't just need to know who is related to who. You also need to know their exact profession, their current GPS coordinates down to the millimeter, and like exactly who they are gossiping with through the wall at this very second.
Which is, yeah, it sounds completely impossible.
It sounds like science fiction. But that is exactly what scientists are figuring out how to do. Only they aren't doing it with people, obviously. They're doing it with the billions of individual cells inside a living body. So welcome to today's deep dive, where we are going to explore exactly how they're pulling this off.
Thanks for having me. And yeah, this really is a holy grail concept in biology, because for decades, researchers have been trapped in a really frustrating compromise.
Right, because we've had ways to track ancestry before, but they always had a massive catch.
Exactly. So we have this technique called single-cell lineage tracing. It's a brilliant tool for tracking cellular ancestry. It basically lets you figure out which cell is the, great-great-granddaughter of an original stem cell.
But the fatal flaw is that to actually read that genetic ancestry, you have to completely dissolve the tissue.
Right. You essentially have to dynamite the city to read the census data. You lose all the geographical context of where those cells actually live.
So you know who they are, but you have no idea who their neighbors were.
Exactly. And then on the flip side of that compromise, we have spatial transcriptomics.
Like SEMA-FIH and things like that, right?
Yep. And these are tools that are incredible at showing us the exact physical location of a cell and, crucially, what genes it's actively using its job description, basically.
But with those tools, the lineage is totally invisible.
Exactly. You have the map, but no family tree at all, which means combining the complete genetic profile, the ancestral history, and the exact spatial location all at the exact same time has just been this massive biological blind spot.
We could look at ancestry, or we could look at geography, but never both. Until now.
Right.
Because today's deep dive is about a groundbreaking paper published in Cell Stem Cell. It details a brand new technology called SPACE-seq. That stands for spatial tracing, enabled by CRISPR-based barcodes and slide-seq.
The name is definitely a mouthful, but the implications are just staggering.
Okay, so let's set a roadmap for you listening. Our mission today is to first look under the hood of the biological machinery that makes SPACE-seq work. Then we're going to dive into a wild case study involving liver cancer.
That part is fascinating.
It really is. And finally, we're going to map the literal mind-bending architecture of a developing brain. So to kick us off, if I'm a researcher, how exactly do I manage to track the GPS, the job description, and the family tree of a single cell simultaneously?
Well, the foundation of this entire system relies on an incredibly sophisticated genetically engineered mouse model. They named the model DARLIN.
DARLIN.
Yeah. And the fundamental challenge of tracing any lineage is that you have to give every single starting cell a unique, permanent barcode, like something stamped into its DNA that it will pass down to every single descendant it ever makes.
So they can trace it back generations later.
Exactly. And DARLIN achieves this using a modified version of CRISPR. Specifically, a doxycycline-inducible Cas9 TDT fusion protein.
Okay, let me pause you right there because the mechanics of this are so cool. Usually when we talk about CRISPR and the Cas9 protein, the classic analogy is that it's a pair of molecular scissors.
Right. Standard Cas9 just cuts.
Yeah, it goes in, cuts the DNA strand at a specific location, and then steps back. The cell panics, tries to hastily repair the cut, and in the process, it usually makes a mistake. It drops a few letters of DNA.
And that missing chunk, that deletion, becomes the unique barcode for that cell.
But there's a mathematical problem with relying just on deletions, isn't there? Like, they are finite.
The options run out very quickly, yeah. If you rely on random deletions, eventually two completely unrelated cells on opposite sides of the body are going to make the exact same genetic mistake purely by random chance.
And if two unrelated cells share the same barcode, your whole family tree is ruined. You'd think they were sisters when they were actually total strangers.
Biologists actually call that homoplasy, and it has plagued lineage tracing for years. You just couldn't trust the data for really large populations of cells.
So how does DARLIN fix homoplasy?
The brilliance of the DARLIN mouse is the TDT part of that fusion protein. TDT stands for terminal deoxynucleotidal transferase.
Wow. Okay. Let's just stick to TDT.
Good idea. So what TDT does is fundamentally change how the barcode is written. Instead of just cutting the DNA and letting the cell make a mistake, TDT is an enzyme that actively grabs random floating DNA nucleotides, the letters of the genetic code, and actively shoves them into the cut site before it heals.
Oh, wow. So it upgrades the Cas9 from a simple pair of scissors to, like, an active molecular pen.
That is the perfect way to visualize it.
It is literally writing new, completely random sequences of letters into the DNA.
Exactly. And because it is inserting random sequences of varying lengths, the barcode diversity becomes astronomically high. It guarantees that the chance of two unrelated cells accidentally generating the same barcode drops to almost zero.
That's incredible. It creates an unerasable, highly complex family crest stamped permanently into the genome.
DARLIN doesn't just write this crest in one random place either. It is engineered to target 3 highly specific genomic locations. They are known as the COL-A, RA, and TA loci.
Why 3 different spots? Just to be safe.
Exactly. It's for redundancy. Think of it like backing up a crucial computer file to an external hard drive and the cloud at the exact same time.
I see.
If the sequencing equipment has trouble reading the barcode at one location, the researchers can cross-validate the lineage using the backups. And the TA and RA locations are naturally very active areas of the genome.
Meaning what exactly?
Meaning the barcodes stored there are constantly being read by the cell, making them very easy for our equipment to detect later on.
Okay, let me just make sure I've got this biological architecture straight. The molecular pen gives us the family crest. We now have a mathematically infinite way to track ancestry. That solves the lineage part of the blind spot.
Right.
But how are we getting the GPS coordinates and the job description? Because we still need the map.
That is where the second half of the technology comes in, the SPACE-seq part. Specifically, they use an upgraded version called SPACE-seq.
Okay, how does that work?
To understand this, imagine a small circular surface. The researchers call it a puck. This puck is densely coated in millions of microscopic beads.
Like tiny little pearls.
Yeah, but microscopic. Each bead is about 10 microns wide, which is roughly the size of a single animal cell.
So it's an incredibly fine microscopic grid.
Exactly. And tethered to every single one of those beads are millions of DNA probes. Crucially, every single bead has its own unique spatial barcode. The researchers know the exact mathematical coordinate of every spatial barcode on the puck.
Okay, I'm tracking.
Now, when you take a painfully thin, frozen slice of organic tissue and lay it flat onto this puck, the cells basically burst open. The messenger RNA from those cells, which, remember, is the active instructions the cell is currently using, spills out.
And it binds to the probes on the bead directly underneath it.
Exactly. And it binds because those probes are designed to grab onto something called a poly-A tail. Basically, whenever a cell reads a gene to perform a function, it slaps a long string of A nucleotides at the end of the message.
So the bead acts like molecular Velcro for those tails.
Yes, molecular Velcro is a great analogy. It pulls down the entire active transcriptome. That's the complete list of every single gene that cell is currently firing.
And that gives us the job description.
Right. But here is the genius stroke of SPACE-seq. Normally, the DARLIN ancestral barcodes wouldn't get caught by that molecular Velcro. They're just structural DNA.
Right, they wouldn't have the poly-A tail.
Exactly. But the DARLIN mouse is engineered so that its ancestral barcodes are actually expressed as messenger RNA, complete with their own poly-A tails.
Oh, that is so clever.
Yeah. During the laboratory amplification step, SPACE-seq uses a specialized primer to ensure those rare ancestral barcodes are pulled down and amplified right alongside the massive pile of normal cellular job descriptions.
That is incredibly elegant. The cell is literally tricked into broadcasting its own family crest right alongside its daily to-do list. And the microscopic bead catches both of them at the exact same GPS coordinate.
It really is a marvel of engineering.
Okay, let's do a quick mini recap for you listening, just to cement this before we move on. Number 1, DARLIN provides the ancestry barcode via CRISPR insertions, like a molecular pen.
Yep.
Number 2, SPACE-seq provides the geographic map and transcriptomic job description using those 10-micron Velcro beads.
Spot on.
And number 3, SPACE-seq marries them together, capturing both the family crest and the job at the exact same physical location.
Which transitions us perfectly from the realm of, theoretical engineering to actual practical application.
Yeah.
Because a tool like this is only as good as the biological mysteries it can actually solve.
Right. What do they actually use it for?
The researchers decided to test SPACE-seq on one of the most notoriously complex and chaotic environments in the human body, a solid tumor.
And they focused on a specific type of liver cancer. Right. Intrahepatic cholangiocarcinoma, or ICCA.
Yeah, it is a very aggressive cancer of the bile ducts within the liver. To study this in the DARLIN mice, they used a technique called hydrodynamic tail vein injection.
Okay, wait, what is that? Tail vein injection.
It sounds intense, and it kind of is. They introduce a large volume of fluid containing genetic tools, specifically cancer-driving mutations like NRAS G12V and a P53 knockdown into the bloodstream.
In the mouse's tail.
Right. And they do it so rapidly that the sheer pressure forces the tools directly into the cells of the liver. It's a very fast, reliable way to induce these tumors. Within 8 weeks, the mice developed multifocal liver tumors.
Okay, so now they have the tumors to study. And this is where the SPACE-seq data completely flips conventional wisdom on its head.
It really did.
When they looked at the tumors, they checked the ancestral barcodes to see where the cancer came from. And they found that within any individual tumor nodule, over 95% of the cancer cells carry the exact same dominant family crest.
Meaning they are completely monoclonal. That entire massive tumor mass sprouted from one single unlucky starting cell that went rogue.
Okay, wait, hold on. This is where my intuition just hits a wall.
Okay, what's the hang up?
If every single cell in that tumor nodule is the direct descendant of the exact same great-grandfather cell, they all have the identical mutated DNA.
Correct.
So shouldn't their DNA force them to behave the exact same way? Like, how can a tumor be this chaotic and adaptable if it's literally just millions of copies of the exact same cell?
That is one of the most hotly debated questions in oncology today. And SPACE-seq provided a definitive answer. It proved a phenomenon called intraclonal transcriptional diversification.
That's a lot of syllables. What does that mean in plain English?
It means that even though the cells share the exact same genetic hardware and the same ancestral barcode, they dynamically shift their software into completely different functional states.
So the tumor acts less like a photocopier just making identical clones and more like a society.
Exactly. When they looked at the job descriptions captured by the spatial grid, they didn't just see one type of cell. Some of the tumor cells were acting like highly specialized experts. They were expressing genes like HR2, which is responsible for producing mucus.
So they were behaving almost like normal, mature bile duct cells.
Right. But sitting right next to those specialists, carrying the exact same family crest, were cells that had entirely regressed.
Regressed how?
They were expressing primitive genes like SOX9 and NCAM1 that pushed them into a dedifferentiated stem cell-like state. This makes them incredibly resilient to stress and chemotherapy.
Wow.
And completely independent of that, you had some cells acting as the engines of the tumor hyperproliferative cells running their division genes, like CCND1 and Ki67 constantly, while others just sat totally dormant.
From one single ancestor, the tumor actively diversifies its own population. You get the specialists, the aggressive stem-like cells, the fast dividers, and the sleepers.
It's terrifying, but also incredibly sophisticated.
Why does this completely shatter the old paradigms of how we view cancer?
Because for a long time, the dominant theory was the cell of origin model. The assumption was simple. If we biopsy a tumor and it looks mostly like primitive stem cells, we assumed the original mutation must have happened in a stem cell.
Oh, I see. And if the tumor looks like mature, differentiated tissue, the mutation must have happened in a mature cell.
Right. But SPACE-seq provides undeniable spatial evidence that this heterogeneity is driven by tumor plasticity. The tumor is actively assigning different roles to its descendants in real time to ensure the whole mass survives.
It's an active survival strategy, not just a rigid genetic destiny. But the tumor isn't executing this strategy in a vacuum, right? It exists in a neighborhood.
Yes, and this is where we zoom out from the malignant cancer cells to look at the non-malignant tissue surrounding them, the tumor microenvironment.
Because the neighborhood is just as crucial as the tumor itself.
Absolutely. SPACE-seq allowed researchers to look very closely at a specific type of neighbor called hepatic stellate cells, or HSCs. In a healthy liver, these cells are quiet and peaceful. Their main job is just storing vitamin A.
But when the liver senses injury, or when a tumor starts growing, these cells wake up.
They become highly active, and they start pumping out extracellular matrix. Genes like VIM and COL1A1 go into overdrive. Essentially, they produce massive amounts of scar tissue. This process is called fibrosis.
And when the researchers looked at the spatial map of these HSCs, they noticed something incredible. They saw massive clonal expansions, meaning these scar tissue-producing cells were rapidly dividing and multiplying, but only in the immediate physical vicinity of the tumor.
It was highly localized.
The tumor was somehow manipulating the peaceful neighbors into building a fortress around it.
Right. But figuring out the exact chemical conversation happening between the tumor and the neighbors required some intense computational mathematics.
Why is that?
Well, remember those microscopic beads on the puck? They are 10 microns wide. Sometimes a bead ends up sitting right on the physical border between a tumor cell and a neighboring HSC.
Oh, I see. So the molecular Velcro catches the active genes from both cells at the exact same time. It's like having a microphone recording two different people shouting over each other.
That's a great way to think of it.
Mm-hmm.
To untangle that audio, so to speak, they relied on a sophisticated algorithm called robust cell type decomposition, or RCTD.
Okay, RCTD.
Mm-hmm.
How does that untangle the shouting?
Think of it like analyzing a blended smoothie. The algorithm knows the exact molecular recipe for a pure tumor cell and the exact recipe for a pure HSC. It looks at the mixed genetic data on that specific bead and mathematically calculates, okay, this signal is 60% tumor and 40% HSC.
Oh, wow. So it mathematically separates them?
Exactly. And once they separated the signals, they layered on another framework called CSide, which allowed them to isolate exactly what the tumor was saying without being confused by the neighbor.
Yeah, and what was it saying?
When they did this, they uncovered a vicious bidirectional feedback loop. The expanded HSCs start furiously producing structural proteins. They are building the physical scaffolding, the scar tissue that the tumor needs to grow.
And in response to the scaffolding being built, the tumor cells located physically closest to the HSCs radically alter their behavior.
They drastically shut down the production of a specific gene called selenop.
Let's unpack selenop because this is the smoking gun of the whole study. Selenop is a protein that carries selenium throughout the body. In the liver, it acts as a powerful, natural anti-inflammatory brake.
It keeps things calm.
Right. But at the exact same time the tumor cells shut down this brake, they aggressively turn on genes that drive rapid cell division, like ANCUS2 and Me9.
The mechanism is a perfect storm. The tumor shuts off the anti-inflammatory brakes by dropping selenop levels. The lack of selenop causes the neighboring HSCs to panic and expand even more.
And the expanded HSCs build more scaffolding, which allows the tumor to grow faster, which further drops the selenop. It is a continuous cycle of corruption.
It's brutal. But the researchers didn't just passively map this corruption. They used this information to launch a counterattack.
Yes, they actively intervened, right?
They did. They went back to the genetic tools they used to induce the cancer in the first place and modified them to artificially force the mouse tumors to continuously overexpress human selenop.
They basically hardwired the anti-inflammatory brakes to stay on, no matter what the tumor tried to do.
Exactly. And the results were stunning. The heavy fibrosis, that scar tissue fortress, shrank significantly.
The physical size of the tumors was reduced too. The localized runaway expansion of the neighboring cells basically came to a halt.
By understanding the spatial conversation, they found the exact sentence needed to stop the tumor from manipulating its environment.
That is just incredible. But this works in humans too, right? It's not just mice.
We have strong evidence it translates, yeah. When the researchers cross-referenced these findings with large human clinical databases, specifically looking at the TCGA-CHL dataset for human bile duct cancers and biopsies from severe fatty liver disease, the data perfectly aligned.
Wow.
In human patients, higher levels of selenop expression strongly correlated with lower levels of liver fibrosis.
Okay, so time for mini recap number 2. Let's synthesize this journey so far. Number 1, we've seen how a single mutated cell can spawn a massive, dynamically changing society of tumor cells. They aren't all the same.
Exactly. They have massive plasticity.
Number 2, we've seen how that tumor actively manipulates its healthy HSC neighbors into building a scar tissue fortress.
The neighborhood watch turned bad.
Right. And number 3, identifying the specific chemical signals, like the loss of selenop, gives us a roadmap to potentially halt that corruption by turning the brakes back on.
That's a perfect summary.
But, if a diseased tissue can organize a chaotic neighborhood this effectively, it raises a really fascinating question. How does a healthy body manage to build a perfect neighborhood from scratch?
That question brings us to the final and perhaps most visually beautiful application of SPACE-seq in this paper. Moving entirely away from cancer, the researchers used the technology to trace the foundational architecture of embryonic development.
They wanted to see the invisible rules that govern how the brain and the liver actually physically construct themselves.
Right. They started by mapping the forebrain of a mouse. specifically tracking development from a tiny embryo at day 10 all the way to a fully grown adult.
And the spatial maps they generated of how the clones expand are fundamentally different depending on what part of the brain you look at.
Oh, completely different. If you look at the neocortex, the outer layer responsible for higher order thought, the clones grow like an elevator shaft.
An elevator shaft. How so?
When a progenitor cell divides, its descendants stack vertically on top of each other, forming these rigid radial columns.
Okay, so straight up and down.
Right. But if you look at the hippocampus, which handles memory and learning, the architecture completely changes. The clones grow like a hallway. They spread out tangentially, sliding past one another to populate the space horizontally.
To really visualize this, the researchers used software to stitch together dozens of incredibly thin two-dimensional slices of the brain into a fully cohesive 3D map.
And when they rendered the hippocampus in 3D, they found an unbelievable level of geometric discipline. The hippocampus has two main structures. One region looks like a curved horn, called the CA region, and nestled inside it is a region shaped like a sharp V, called the dentate gyrus, or DG.
And the spatial data revealed that the cellular families absolutely respect the borders of these shapes, right?
Yes. A clone born in the horn region almost never crosses the invisible boundary into the V-shaped region. But even more fascinating, within the V-shape itself, the families exhibit perfectly mirrored symmetry.
It's wild. The clones populating the top arm of the V, the suprapyramidal blade, are a perfect geometric mirror image of the clones populating the bottom arm, the infrapyramidal blade.
It implies that there are incredibly strict, invisible chemical force fields guiding these cells into perfectly balanced, mirrored positions as the brain develops.
We've always known the brain is complex, but seeing the geometric precision of the family tree is something else entirely. But then they applied this exact same developmental mapping to the liver. And the liver uses a completely different set of rules.
Yeah, the liver is wild. When mapping how the lobes of the liver form, SPACE-seq uncovered what the researchers call a dispersion window.
A dispersion window?
Yeah, so if they tagged the ancestral stem cells of the liver, the hepatoblasts, very early, at embryo day 10, the resulting clones scattered wildly.
Like how wildly?
A single family crest would end up populating multiple, completely separate lobes on opposite sides of the liver, massively intermixing with completely unrelated cellular families.
But if they waited just 48 hours to tag the cells, labeling them at embryo day 12, the behavior changed entirely.
The cells completely locked down. By E12 to E15.5, they only divided locally, staying strictly within their own microscopic neighborhood.
Now, if I'm trying to build a complex structure, this seems incredibly counterintuitive. If you're building a house, you don't randomly scatter your bricks across 3 different zip codes for 2 days and then suddenly decide to start building locally.
It sounds crazy, yeah.
Why does the liver aggressively scatter its cells far and wide early on, only to abruptly lock them down?
It actually makes perfect sense when you view it through the lens of evolutionary risk management. The body is balancing 2 competing necessities: genetic flexibility and structural precision.
Okay, unpack that for me.
Think about the risk of building locally from day 1. If a single early stem cell has a fatal genetic defect and it only divides locally, an entire lobe of the liver will be built from defective tissue. That lobe will fail.
Ah, I see. By forcing the cells to wildly scatter and migrate during that early E10 window, the liver acts like a deck of cards being thoroughly shuffled. It intermixes the families.
Exactly. It diversifies the genetic makeup of every local area, acting as a buffer. If one family line has a defect, it is surrounded by healthy cells from different lineages that can compensate.
It's an evolutionary insurance policy.
Exactly. And once that genetic insurance policy is distributed across the whole organ, the E12 lockdown occurs. The cells stop migrating so they can focus on constructing the incredibly precise, complex vascular plumbing required for the liver to actually filter blood.
Because you can't build intricate plumbing if the pipes keep moving around.
Right. You need local stability for that.
It's a hidden, fundamental rule of organ construction that balances chaos and order. And it was entirely invisible to us before SPACE-seq allowed us to read the map and the family tree at the same time. Let's do our final mini recap here.
Sounds good.
Number 1, brain clones grow radically differently depending on the region, vertical columns in the cortex, horizontal hallways in the hippocampus.
Yep.
Number 2, early liver development features a wild dispersion phase to shuffle the genetic deck before settling into local growth, all to prevent catastrophic local defects.
Spot on. The ultimate value of this technology isn't just that it produces beautiful, high-resolution biological maps, though they are beautiful.
They really are.
But it provides us with the dictionary to read the invisible rules of how bodies build themselves and exactly how diseases hijack those rules. By understanding the spatial and ancestral context of a cell, we are unlocking the potential for highly targeted, state-specific therapies.
Right, like we don't just see that a liver is fibrotic. We see exactly which tumor cells are dropping their solanop levels to manipulate their neighbors.
And because we know the exact mechanism, we can target that exact pathway to stop the corruption in its tracks.
So as we wrap up this deep dive, you've just heard how the cells inside of you carry a hidden, unerasable history of exactly where they came from and who their neighbors are. Every single cell in your body right now is gossiping, adapting, manipulating, and relying on its neighbors.
Following intricate geometric and evolutionary rules that were established when you were just a microscopic embryo.
Exactly. If scientists now have the tools to read a cell's entire ancestral history, its current job, and its spatial context down to the exact millimeter, what happens when we figure out how to rewrite that history in living tissue? Could we rewrite the geographic instructions of a tumor to make it dismantle its own fortress? Could we reset the neighborhood cues to reverse aging or stop cancer before it ever has the chance to form? Think about the complex microscopic societies interacting inside your own cellular neighborhoods right now. We'll leave you with that.